Before I start my lecture, I should place acknowledgement to all my co-contributors, Dr. Neeraj Kumar, Yogesh Kumar, and Sailesh Tripathi from Indian Institute of Pulses Research, Sushil K. Chaturvedi, sir, who is Dean Research at RLBCAU. Now, when we start looking at where pulses stand, basically in Indian and world scenario, let me say that India is one of the largest pulse producing country in the world. It is also one of the largest importer of pulses, and the parody is that it is also the largest consumer of pulses. So much so that the largely vegan population continues to import from 3 million ton to 10 million ton of different kinds of pulses over the years. Now, saying that, we started with 13.38 million tons, and till last year, 24.5 million tons, there has been an impressive growth of 106%, the largest in case of any other crop, followed by or nearly completed by millets. It's a big leap of India towards attaining self-sufficiency. And when you start looking at chickpea itself, it is one of the major pulse in India. Of course, it is the second largest pulse in the world after dry beans. We cultivate it in a 14.8 million hectares of area, published statistics of 18.1 million tons. It's roughly staggering around the same thing with a productivity potential of 1.1 ton, which is, of course, way below the average of certain of the leading countries. But then we need to understand that chickpea as a production scenario is not taken as a sole crop, as is done in case of Canada or is in case of Australia or even in case of the Middle East. It is basically a two crop system rotation, either a soybean followed by chickpea that we take up, or it is basically the kharif crops followed by chickpea that is taken up in the northern countries. So we need to understand that per day productivity is higher, but there is almost a good amount of production potential increase and average productivity that can be achieved. Nevertheless, when we speak of pulses per se, we see that the production has increased by 178% in case of moong bean, 125% in case of chickpea, 90% in case of urad, 51% in case of pigeon pea, and 34% in case of lentil. Chickpea almost contributes to 50% in the total pulse production, whereas it's closely followed by pigeon pea, moong, urad, lentil, and others. We have a large amount of states that grow chickpeas, which is the main crux of my topic today. However, MP leads the production potential of this, followed by Maharashtra, Rajasthan, where more than 95% of the chickpea production of the world occurs. Now, if you're looking at pulse production in India, you can very clearly see that somewhere between 694 kgs of total pulse production per hectare per unit area, we have increased to 600 and 892. At the same time, we brought in a near self-sufficiency. Well, a near self-sufficiency that needs to be almost increased, doubled by the same amount, as we will be seeing in the succeeding slides. Dependency of import has greatly come down. Now, the parody is every year, one or the other pulses become scarce, like it was chickpea last year. Before that, this year it is what we call as arhar or pigeon pea. Before last year, it was basically the lentils that were in shortfall. So pulses as a basket, one or the other pulses, generally due to the vagaries of the climate, fall down in production, and therefore we become an import dependent on that. This year, that's why the government has increased focus with good amount of self-reliance in chickpea, the increased focus onto lentil, moong bean, and urad bean to try to increase and have a good amount of buffer. Now, if you look at the current population of 1.38 billion, we would be crossing 1.69 billion by 2050. The parent capital availability as such is 52.9 grams from the present 19.3 grams, whereas ICMR recommends 54 grams. So we are having about 28 million tons, and we require to be increased it to almost, as I said, 52 million tons. From that of somewhere around 24, 25, there was a small dip after this. We need to now increase to a huge amount of 52 million tons. That too, from decreasing area, from decreasing inputs, from pulses being pushed every time away towards more marginal or submarginal or semi-arid tropics. Now, if you look into the pulse production per se, you can understand that gram stands as a king, both in terms of contribution, both in terms of area. However, still a large can be achieved. In our Bill and Melinda Gates Foundation program that we are carrying out at IRI, IIPR, and other group of coordinated centers, we could identify that we had achieved a growth target of 1% genetic gains. However, we intend to increase it to 1.5%. That is a whooping, almost increasing it by 200 times more to achieve a 50% more of genetic gains. We are looking at the modern approach of genomic selection and other sorts. Now, if you start looking at the state-wise contribution, you find that more or less the statistics remains same, where the percentage to all India comes surprisingly from three states of Maharashtra, Rajasthan, and Madhya Pradesh. Of course, Telangana is very fastly approaching, and similarly Gujarat and this thing. And when you start looking at state-wise production percent to all India, when you start looking at here also, you can look that Maharashtra, Madhya Pradesh, and Rajasthan contribute a large. Now we have certain states which are always higher than the all India average, like Gujarat, Madhya Pradesh, and we have certain pockets within Telangana as well as in Maharashtra and Gujarat, which are much above the world average production or world's best production potential. So we need to look at the gaps, the good agricultural practices that actually lead to such production. Now, one peculiar thing that has happened with chickpea when the advent of green revolution has come, that is where it was a very large area in case of northern India, whooping 70%, has now reduced whooping to 18%, with central India and south India increasing both their acreages and production. One is Heliothis, one is the Chara Prata of the northeastern plain zones. The third is with the increased irrigation facilities, whether it is Ganga Nahar or whether it is the Rajiv Gandhi irrigation canal or the Jawaharlal Nehru irrigation project. Most of the farmers started to going towards the cereal crops like rice and wheat, pushing out under chickpea. So if you want to repatriate this lost area, because chickpea takes very less amount of what we call as the requirement of fertilizers, it also has the benefit of fixing of atmospheric nitrogen, helping in a way large of biomass supplementation to the soil by way of leaf fall and other things. Apart from rhizobium basis of nitrogen fixation, we have an urgent need to develop what we call as fertilizer responsive determinate cultivars. That's what IRA is basically working. Now, what good thing that has happened in case of chickpea is there are two factors in any crop production. You need to understand this. One is varietal replacement ratio, another is seed replacement ratio. Due to the policies of DAC, that of the seed unit at ICAR as well as IAPR, the coordinating units, as well as direct character IAPR, we could find that there is an excellent varietal replacement ratio. See, somewhere from 39.1, 65.4% of the varieties are those varieties which are of the last 10 years released. Maybe because you have only benefits for seed indents, but still some old varieties are ruling. Why? Maybe they have yield advantages, may have their local demand. So these needs to be looked out. In lentil, it's a whooping 74. In field pea, it is almost 80%. But now when we come to seed replacement ratios, we find that it is around, which was around 30%, it has now come to around 50 to 60% in most of the pulse crops. That means if you look on other way around, the farmer saved seed or farmer exchanged seed is still a major player in case of seed production, in case of most of the pulse crops. And this needs to be very carefully looked into, and maybe the FPOs, maybe the seed production agencies have to gear up, particularly those varieties which are about 5-year released. So there was a good growth, as we see in basically in case of pulses in general and chickpea per se. The pulse mission mode has made a very good dent in the production of pulses and chickpea. The availability of high yielding varieties, kudos to the IAPR and the project coordinating unit that of different environments and the growing different target production environments. Most important thing I consider is empowerment of farmers with knowledge of improved cultivars and production technologies. The area expansion in non-niche areas, that is the non-traditional areas like rice fallows, spring summer season, and other non-traditional areas. There has been luckily a favorable government policy, irrespective of the governments at the center, since almost 1990s in favor of pulses. Now, a good amount of short duration pulses have been developed thanks to Dr. Puran Gaur and the Southern research teams like Jackie 9218, JG14, ICCV10, which is one earlier. And of course, we have Pusa Manav that has come off recently. Similarly, we have Pusa JG16 and Pusa 10216, which are highly drought tolerant varieties. Now, the availability of a short duration pulse, the availability of irrigation source at the critical stage, and third, the availability of warehousing facilities in case of Andhra Pradesh and Telangana have brought a renaissance in case of chickpea production. Perhaps other states need to evaluate this. Availability, as I said, of fusarium wilt resistant varieties. For example, Pusa Manav has been tipped off as a super chickpea variety with an average production potential of 3 tons per hectare, unheard, and with a production potential of 4 tons, and most of the farmers taking almost 4.5 tons per hectare. It is a marker-assisted bred variety where all the QTLs for the fork races have been pyramided into one cultivar. We could develop Ascochyta blight resistant varieties, particularly from PAU like PBG7 and PBG5. The heat tolerant varieties still play a great role, JG14, RVG202, RSG888. The drought tolerant varieties that have been recently developed, which are occupying large areas in Gujarat, in case of Latur and those, Pusa Chickpea 10216, Pusa JG16. For the Northwest Plain zones, that it has been for the first time released, Pusa 4005. One of the biggest problems that the farmers have been facing is the availability of labor, particularly at the harvesting time in central India, where the harvesting coincides with the wheat harvesting. So people were looking for mechanical harvesting varieties, and we are happy that Pusa 3062, that is Pusa Parvati, is the first mechanical harvesting variety for the biggest chickpea cultivation zone, that is the central zone, followed by large number of varieties, particularly Jayalakshmi's NBEG 47, Phule Vikram RVG 204. Now, what are the various seed production strategies of chickpea? Now, if you look, there are different breeding methods when you start basically looking from plant introduction to that of a pure line selection to mass selection, pedigree method, bulk method, single seed or backcross descent, to the special methods of mutation breeding, ploidy breeding, transgenic breeding, and molecular breeding. Now, chickpea is a peculiar plant. It has a taproot which is deep and strong and is a herbaceous annual. One of the specific characters of it is the flower type. The flower belongs to leguminosa, therefore, you have a specific flower character with long keel and that which acts as a covering to prevent the pollen from being exposed. And it is clistogamy, that means by the time the flower opens, already fertilization does occur. So we have two types of chickpea, very well known. The bees colored, which have an export value, as well as used basically for culinary, that is the Kabuli types. They're used for culinary purposes because these have greater amount of the sweet sugars, the raffinose family sugars, and hence have a good taste. And the Desi types, 80% of the chickpea production generally is of the Desi types. Basically, this is used for flour formation. Flour is used in a huge amount of things, from that of making savories to that of different dishes, whereas this basically used as culinary. People want the taste of this, but they prefer the color of Desi. Therefore, what they basically do, add a tea bag when they are actually cooking them or blanching them. So basically, what we are looking for is to produce less gas-causing chickpeas, the Kabuli bold types, and we are working on reducing the raffinose family oligosaccharides, which are basically the gastulence factors in case of chickpea. Now, the simple thing is it is a hypogeal germination, occurs in 7 to 15 days, which as a seed producer, you need to take care of. It has about 15 degrees to 25 degrees as its best growth maximas, but it can grow till almost what we call as 38 to 38 degrees and as low as 10 degrees. We have certain cold tolerant varieties which can come off at even at 8 degrees. Of course, this is with respect to either cessation of growth and staying as such, or carrying on the vegetative phase at a lower scale. Pollination and fertilization ideally occurs at 25 to 28 degrees. As I said, it's a papilionaceous flower and is clistogamous in nature. Therefore, anthers dehisce well before 24 hours before the bud pollination. Generally, about 10 to 15 days, the pod starts forming. Just like other legumes, which includes soybean itself, the pod forms first and then the seeds then start developing. So we have distinct stages well identified in soybean and applied in chickpea: R1, R2, R3, R4, R5, R6, R7, and R8. R1 to R3 are the pod expansion stages. R4, the seed as an immature embryo starts showing in its piriform conditions, grows to marble size, and then grows to maturity. Now, the sowing time generally chickpea is for commercial productions, is post rainy season, or in India we call it as Rabi crop. So basically it takes place in October and November. And then in late sowing, some people almost take it to late till December 15th. But one of the biggest problems is the moisture stress and high temperature at pod filling. Now, I said we have developed specific varieties where QTLs for drought tolerance have been mobilized from ICC 4958 with a group of breeders from Ikrisat, Varshne, Manish Rurkiwal, and the group, and at IRA, IAPR, where we could mobilize these QTLs into some of the most prominent lines. Chickpea being a self-pollinated crop, so therefore huge amount of isolation distance is not required. It's basically 10 meters for foundation, and just 5 meters for certified is more than sufficient. This slide in a gist provides the hypogeal type of germination, and you have the first few cotyledonary seeds and the vegetative phase. Pre-flowering stage is one of the most critical stages for water stress, and the second is the pod development stages. At the time of flowering, the amount of nodules that are present are maximum and slowly start decreasing. As I said, it is a peculiar leguminoid flower shape with the standard petal, standard keel petal that basically protects the inside anther and other floral structures, and you have wings that are basically present. You have 10 anthers, 9 plus 1 in a didelphous conditions. So pollination needs to be done before 10 or 11 when temperatures start slowly rising. It's a bit difficult, but then pushing open both the keel petals would actually expose the inner anthers. And with one swish of forceps, you can just pull out them holding that, so that only the stigma is present. And then once temperatures rise, as I said, when you start looking of all the stages R1, R2, R3, R4. So till this time, it's a pod development stages. So R4, you have miniature embryos that are developing. R4, R5, R6, R7, R8. So when the pod color turns into yellow, it indicates it's time to mature. And then we try to harvest and have 1 day of curation that most of the farmers do in case of chickpea. Now, it is very well understood that you need to have a pH of about 6 to 7, that is most ideal. Saline soils or soils with high water table are not suitable for chickpea after an initial irrigation. The soil is well turned to make it into good ply conditions, so that there is loose tilth and good drainage. So this, as I said, we do it by pre-sowing irrigation. So generally chickpeas are not sown very deep, ideally roughly 2 inches depth is ideal. Now spacing, line sowing always facilitates better intercultural operations. Now you need to understand that farmers are quite innovative. Though generally we say that 30 centimeters row to row and 10 centimeters between plant, that brings about roughly 3 lakh plants per hectare is sufficient. Now, some of the very progressive farmers like Gulab Singh Lodhi or Anit Mangul, or we have very good farmers, even in case of Ethiopia, who are with Asnan Fakare or Paul Kimomoto in Kenya. These farmers have developed a way of BBF, that is broadbed and furrow system, where just using 6 to 8 kgs per acre, these farmers actually dibble at almost what you call as 5 feet distance bed and almost 14 inches between the plants or the well spreading manner, or 10 inches between others, and then therefore reduce a huge amount of seed rate. However, the recommended from IAPR and what we say are those seeds which are less than 20 grams, like JG 315, 60 kgs per hectare. The medium ones to 90, to very large and extra large about 150 kgs per hectare. Irrespective of the seed, we always say that go for seed treatment. We have two groups of farmers who are here, or production people. One, the organic, and the one who follow the chemical ones. For the ones who take up organic, we do say give 2 grams of thiram and 1 gram of carbendazim per kg seed to reduce wilt, followed by, just before sowing, we give rhizobium treatment. Those who are taking up organic cultivation can go by trichoderma viride application, also with rhizobium and PCB as a mixture. Fertilizers, generally it is 20, 40, 20 that is recommended based on soil testing. I certainly advise you. We miss out sulfur and we say that 20 kgs per hectare, basically of zinc sulfate, should serve as an ideal source, most of the pulse crops. And if you are in a soybean chickpea belt, please understand that soybean is a beautiful uptaker of molybdenum. Therefore, you need to apply molybdenum. Or if you're taking maize, then it exhausts most of the micronutrients. Irrigation, as I said, two irrigations are critical. One at branching just before flowering. At the time of flowering, if you give irrigation, what happens is chickpea be inherently is determinate perennial type inherently. Therefore, it sheds flowers and picks up beautiful vegetative growth, and therefore then the flowering becomes limited to the top terminal portions than from starting from base, and therefore yields are very much reduced. So pre-flowering one and at the time of 50% podding is highly recommended. We expect the fields to be weed free till 40 days after sowing. So chemical people can go about with applying pre-emergence herbicides like pendimethalin, commercially stomped, or using plastic mulches. We have farmers who are actually using even drips for growing chickpea with mulches. And these people like Kanit Mangul, I always take the example, or Prithvi Solanki from Madhya Pradesh, they take as high as 6 tons per hectare, something recordably insane that I have seen. Now, there are good amount of diseases. As I said, you have fusarium wilt, and for a chickpea variety to be released, it has to be resistant to fusarium wilt, collar rot or dry root rot. You have package of practices, as I said, seed treatment, drenching. But what always I advise is basically to look into ways where you can have crop rotations, a crop holiday for chickpea, because or any legume per se in the same field, because the soil borne pathogens which have a broad spectrum of legume crops and pulse crops start building in the soil, be it rhizoctonia, be it fusarium, be it even dry root rot fungus, and be it even termites. So you need to give good a crop holiday once in a while, follow crop rotation. Third most important, deep plowing in summer, of course, a difficult thing in case of BC soils, black cotton soils. So you need to look out so that the deep plowing exposes the inner layers of the soil, the particular insects and the larval pupae to hot summers, as well as the spores of fungicides killing them. Flushing out of weeds by having a pre-sowing, pre-irrigation before pre-sowing itself, maybe 15-20 days before. Therefore, the weeds germinate and then they're plowed totally. Then, of course, pre-emergence or herbicides or going for mulches. Termites are one major problem, and of course, you need to look out that light irrigations and particularly having chlorpyrifos drips can take them down. We have a cutworm, and we have a package of practices that works. These are some of the insects diseases that are present. A typical fusarium wilt, where if you split open this, what happens is you'll find black tissue. When you look at the dry root rot, the fusarium wilt comes during cold, dry conditions, and this comes under totally increasing temperature. At the later stages, the root becomes almost dried out. The difference between this and the termite is the root is stumpy in case of termites and not so in case of dry root rot. The Ascochyta blight, one of the devastating diseases worldwide, is basically occurring of concentric circles on the pod and stem. We have very good tolerant sources. Still, we don't have good sources for collar rot and botrytis gray mold, and we are trying to manage it or develop and identify genes that are playing a major role for disease resistance. Bruchids are one of the major things in case of storage pests in chickpea. The infestation starts in field. So before harvesting, it is 10 days before harvesting in the pod stage, we generally recommend spraying of some systemic insecticide to prevent the moths from laying eggs after looking at the ETLs, economic threshold limits, that is 5 moths in a light trap that can be trapped. Now, roguing is one of the most important aspect of seed production in case of chickpea. We recommend roguing basically at 3 stages, but at least at 2. That is pre-flowering stage, looking at the leaf type, plant height, foliage color. At the flowering stage, looking at the specific flower color, and at the pod stage, the specific pod characters where it are formed, and of course at the seed stage, but 2 stages are generally recommended. The off types are to be removed. Now the harvesting and threshing, when you're doing for a seed production, we say that please decrease the cylinder speed to 400 rpm instead of the commercial 600 to 700 rpm. At a low thresher speed, it is always recommended. Drum harvesting or hand threshing is good for seeds, but however, when you look into large acreages, it is not possible. Similarly, you need to bring down to the moisture of at least 10% ideal, or at least 12% below for storing in well fumigated what you call a storage structure, seed storage. That's what I said, at 10 to 12 months for short term storage. We have basically the double lined poly bags in which they have to be stored on about at least 5 to 10 centimeters above the ground level with proper basement, and not more than 10 bags that is recommended of stacking. Most ideal would be 6 bags of stacking in well what you call as fumigated room. Now, as we see that there is a basic or nucleus seed which is produced by the breeder, which is 100% genetically and physically pure, followed by the breeder's seed where it is 99% to 100% pure, to that of the seed agencies or the seed commercial seed agencies who are in the front line before the farmers, like the foundation and the certified. The multiplication ratio is about 20, as I indicated earlier also. This is the seed standards in case of foundation and certified seed for chickpea seed production. Now, if you look down from the nucleus seed, which is not a class of seed, but is the basic seed that is produced by the breeder and given to its university or the breeding seed production, maybe national seed agencies for breeder seed production, and you have this chain which should have been told. But you need to understand that at any case, there are not more than 3 generations of certification, after which no certification works. So we have both the public sector and private sector. Whether it is public sector and private sector, it tries to develop a variety, evaluate this variety. This variety evaluation in private sector does on multi-location evaluation, and that they have their own initial evaluation trial, need not to submit, but they need to register this variety. But whatever it is, if they want to produce the seed, then they need to have a protection of it and bring it into notification. So once certification of a variety is done, only certified varieties have a legal status, and with insurance playing a major important role, you need to certify a variety. Now you have a system of seed production in India where this agriculture is a state subject. It's not on the concurrent list, and therefore what happens is the Department of Agriculture and Cooperation and Farmers Welfare, what it does basically is get the indents from various seed associations, farmers seed production agencies, or the Director of Agriculture of various states, or the seed corporations and NGOs. It compiles them and therefore sends it to the apex body at ICR. And therefore, in case of chickpea, it is the ADG seeds who actually sends them to IAPR, through which they have the project coordinating unit, both for Rabi and Kharif pulses, chickpea being Rabi pulses. Professor Sailesh Tripathi, who is also one of the co-contributors for our lecture, looks after this, and he instructs, makes what we call as a seed production plan. So this is given to the amount of seed that needs to be produced by the concerned agencies, which are lifted for breeder seed production and foundation seed production. As I said, at every stage you have what we call as the seed certification agencies looking at least 2 stages of certification. Now we have a process wherein you have tabs or mobiles which actually looks into whether the breeder or the certification people have gone there with their photo and geo coordinates that are basically tagged. So this is what is the type of seed production that occurs. I'm not going much detail into this, but I would like to deal in the next 5 minutes before I close into my lecture. What are the major contrasting things, particularly in case of chickpea? In north, it is excessive biomass is the trait because we are looking at temperature extremities. We don't want long, very long days, but not very short either, because fog is one problem. In January, February, if they're having very low temperatures. In April, May, when they're maturing, it comes into very high temperatures, as you see now. In south, the trait is because we are looking at terminal drought. We need to look how can we improve the biomass because of very short duration. We are looking at 85 to 100 days crop duration and harsh post-anthesis period. In central, we are looking at both terminal drought and the heat. So we require something around 110 to 125 days. You have certain occasional frost and drought. Looking at these things, we need to look that chickpeas predominantly rain-fed with low water use potential, efficiency, and we need to improve this. And that's what we were doing using the drought QTL. We also need to look how we are going to work towards cold, frost, and high temperature in case of north. The declining factor productivity is one of the major concerns, and how are you going to increase it? Why I said declining factor productivity is you apply greater amount of nutrients, chickpea takes, but it does not convert that into yield. So it is not nutrient responsive as happened with post-green revolution of rice and wheat. So you need to identify nutrient responsive chickpea idiotypes, and that is the thrust of the current breeding programs, either at IRI or IAPR or at PAU that we are looking into. Looking at semi-determinate or determinate lines, which can actually have a better nutrient response. And we are also working on having better phosphorus use efficiency. So that is one. Weak research and extension linkage, we are trying to improve upon it within the state departments and the central. This is one of the more major important. As I say, we have a fusarium wilt, which is very major causing. And as I put into this map of various races that are prevalent across the countries, and this becomes one of the classical example of translational research, where we brought in all the yield as well as fusarium wilt resistant fog genes into a single cultivar. It has a whooping advantage, almost with a potential of 4 tons in acre with a grain protein content of 19%. We have a huge amount of lines already in pipeline, which are having very high productivity and will. This is a Wilczek plot, have a great level of immunity. I'm not speaking of resistance. Ascochyta blight is one, and PAU has come down with beautiful cultivars that are having resistance, and they developed the resistance through wide hybridization. Botrytis gray mold is one more area. It's still a gray area, I say, but PAU, IRI, and IIPR were able to bring down the resistance from a wild species. We have one cultivar that has been now identified for release from PAU through this combined program. But as I said before, dry root rot and collar rot, none of the sources are stable. What we get in artificial screening is not showing in field, and in field it is below tolerance. So still all the breeders and pathologists are burning the night's candle to actually find out a stable resistant sources. Once a pathologist gives me, I can identify the genes, but till then, these are still a challenge where all the chickpea community is working. Pod borer, we have a transgenic chickpea, but due to embargo of Biodiversity Authority, we are unable to get into the field. Nevertheless, we have good management practices, both what we call as the integrated pest management practices well worked out, both the biotic as well as what we call as the chemical factors. The farmers need to apply this. Looking at prophylactic sprays after identification of VDLs, I always use this word. 5 moths in a square meter in a light trap. We put 5 light traps in acres field. That works out to be 25 moths in a whole acre area, or it is 3 to 5 egg masses per unit area where you need to take this control. But you need to understand that it is only the first instar larva that you can control, and if it goes to third or fourth, God help us. Salinity is one more major problem. Excellent work by IRI, IIPR, and PAU. We are on the verge of developing saline tolerant chickpea lines. Salinity manifests at the seedling stage. We have CSG 8962, Karnal Chana 1, which is already released as a tolerant variety. Now we have better lines in the pipeline that are coming out. As I said, we try to introgress this drought tolerant QTL from ICC 4958, a germplasm line, into cultivated line. And this is the world's first marker-assisted bred chickpea variety that has been released for drought tolerant for the largest chickpea cultivation zone, that is central India. It is Pusa 10216. This QTL's effectivity is very well seen when it has also been released, as I said, in Ethiopia by the name Kimomoto. Similarly, you have from Kenya that has been basically released. So that's what I say. Whooping, we have 111 quintals in the first year of its release about this variety. Similarly, we went ahead with releasing Pusa JG16, which is much more higher yielding. And then this varieties provide an insurance to the farmers, particularly when it is a little bit late sown. In the late zone conditions, I'm saying these gives 1.8 to 2 tons, which no other variety can match. This is an attractive variety with an yellow seed coated color, highly having more than 2 tons per hectare of field production. We have also released one for Northwest Plain Zone, and with IIPR Samriddhi we have released, that is also a fusarium tolerant dedicated to nation by Honorable Prime Minister of India. Now we need to look at a policy incentivizing, increasing of minimum support price. It's not just minimum support price, but procurement by the government agencies, which occurs in case of rice and wheat, which is not there. Chickpea, it was a little bit done. I hope it continues. Most important, excellent thing that Government of India DAC has done was to create seed hubs. Therefore, whooping from 30% seed replacement ratio, we have gone to almost 60% by developing of farmer production agencies, FPOs, and seed hubs. Creation of biofertilizer and biopesticide units. Then yield demonstrations, which were done individually to that of cluster demonstrations. Maybe imports in my thing needs to be restricted if Indian farmers have to be uplifted in terms of the price, this thing. But of course, policy decisions do matter, that when prices certainly increase very high, you need to buffer it up by bringing down the prices by importing. Grow More Pulse campaign with National Food Security Mission on pulses has really helped a lot, and it needs to look into it. Above all, one of the biggest things that has come is e-market integration that needs to be done. So the impact of pulse research in various crops, not chickpea alone, you can see that we have a plus 5.28 in case of chickpea to that of 0.42 in case of urd bean. In general, when you look at a basket, we have plus 8.43. Nevertheless, how to double this whole to almost 50 tons is the biggest challenge. I think the chickpea community would work out using the policy supports, the innovations in seed systems were bringing in more private players and upscaling of technology. I believe that when this triangle is met, then you have better seed availability and seed production techniques need to be implemented. I thank